In this video, we will explore the fascinating process of fertilization in swine. The structure of the swine female reproductive system consists of vagina, cervix, uterine body for early sperm deposition, uterine horns for embryos implantation, oviduct, and finally, the ovaries. The oviduct is a complex structure includes the infundibulum, ampulla, isthmus, and the uterotubal junction. As estrus or heat approaches, changes begin to occur within the reproductive system. Each ovary develops 6 to 10 follicles, with each follicle containing a maturing ovum. Follicular growth accelerates, driven by the release of follicle-stimulating hormone from the pituitary gland. Ovulation, which is the release of the ova, is then triggered by luteinizing hormone, shortly after estrus begins. Once released, the ova travel to the ampulla. During mating or artificial insemination, semen is deposited into the cervix. Boars can deposit as many as 30 to 60 billion sperm during natural service. Uterine contractions stimulated by oxytocin assist in sperm transport. Oxytocin is released from the pituitary gland in response to the stimuli of mating behavior and copulation. Once inside the female reproductive tract, sperm must undergo a process called capacitation, which takes about 6 to 10 hours. This involves significant physical and biochemical changes. Firstly, sperm shift from a linear swimming pattern to vigorous, hyperactivated movement. Secondly, molecules like cholesterol are removed, preparing the sperm for the acrosome reaction. Fertilization occurs primarily in the ampulla of the oviduct. The sperm release an enzyme called hyaluronidase from their acrosome, enabling them to penetrate the corona radiata, the outermost layer of the ovum. Next, they reach the zona pellucida, where the acrosome reaction continues, breaking down this protective layer. Finally, the sperm reach the plasma membrane. Here, the membranes merge, allowing the sperm's nucleus to enter the ovum. The genetic material from the sperm and ovum then combine, forming a single cell known as a zygote, which begins dividing and developing into an embryo. After fusion, the ovum releases cortical granules, which modify the zona pellucida to prevent other sperm from entering. This ensures that only one sperm fertilizes the ovum. However, fertilization isn't always perfect. In swine, polyspermy, where multiple sperm fertilize the ovum, can occur. This is more common during in vitro fertilization, as the high concentration of sperm used exceeds natural levels. Additionally, the swine's zona pellucida may not always harden effectively after the first sperm entry, allowing other sperm to penetrate. These abnormalities in fertilization generally resulting in embryonic death because excess male centrosome delivered into the ovum. Embryos are unlikely to survive beyond the earliest stages of development. And that's the remarkable journey of fertilization in swine.